Dr. Avneet? Dr. Avneet?
Yes, a very good morning, sir.
Good morning, Avneet. How are you?
I'm good, sir. How are you?
I'm fine. Can I see your video once?
Yeah, actually, I'm in clinic, so if that's okay.
Okay, all right, no problem. No problem. But
I'm
if
in
you want,
clinic.
you can on when you speak, all right? When you speak, you can on
Can
it.
you please repeat, sir?
When you speak, then you on it.
I can't hear you clearly, sir. Actually,
No.
there's a lot of disturbance.
When you speak, you on your video.
Okay. So should I introduce sir right now?
No. I will first speak 1 or 2 lines.
Right.
And then you speak.
Swarnijit Singh, sir.
Yes, sir.
Now you can start.
All right. Dear Dr. Deshmukh, dear Dr. Manjeet Singh, and dear Dr. Joseph Bhagyaraj, and all the participants of this event. Today we have an eminent professor and scientist, Dr. P. Joseph Bhagyaraj, with us. He has done a lot of work on microbes, biotechnology, and biodiversity. I thank Dr. Joseph for accepting our invitation and coming on this great platform. Now I call upon Dr. Avneet Kaur of General Medical College Hospital, Sector 32, for the welcome. Dr. Avneet.
Very good morning to everyone and a very warm welcome to our esteemed and renowned chief guest. And the accomplishments and achievements of our chief guest are exemplary and outstanding. So I'm just going to present a few of his achievements. He is the NASI Honorary Scientist and Chairman of CNBRCD, Bangalore. He holds a PhD from University of Agricultural Sciences, Bangalore. He also has postdoctoral experience at New Zealand, Australia, UK, and USA. He is a fellow of Indian National Science Academy, National Academy of Sciences, National Academy of Agricultural Sciences, the National Academy of Biological Sciences, the Society of Agricultural Biotechnologists, Indian Phytopathological Society, and Association of Microbiologists of India. He's also been proudly awarded with national awards like S.R. Vyas Memorial Award, Shom Memorial Award, Dr. G. Rangaswamy Memorial Award, Dr. R.Y. Roy Memorial Lecture Award, Dr. Natarajan Memorial Lecture Award, and also a recipient of Distinguished Asian Mycologist Award 2019 from Asian Mycological Association and several Lifetime Achievement Awards. He has written 418 research papers, 112 review articles, 11 books, 14 popular articles, principal investigator of 36 research projects, and also is currently the PI of 3 research projects. He's also an advisory committee member of ICAR, DST, DBT, CSIR, UGC, NBA, Rubber Board, Spices Board, IFS Sweden, EU, FAO, and UNEP. He's also taught UG and PG courses in microbiology for the past 50 years. Currently, he's guiding 2 PhD students. He was also invited by the European Commission to author the final chapter on mycorrhizal fungi for the Global Atlas on Soil Biodiversity. So, honoring his contributions, a new AM fungus has been named as Glomus Bhagyaraji. It's an honor to have you, sir. I would also like to introduce Dr. Arvind and Dr. Swaranjit Singh, who's the man behind the entire event today. A very good morning to all of you, sir.
Now I call upon Dr. Joseph Bhagyaraj to present his excellent talk.
Okay, thank you very much. And so Dr. Deshmukh, Dr. Swaranjit Singh, I'm thankful to you for inviting me to give this talk. to the Microbiologist Society India. And I'm really very happy to do this, because always take pleasure in taking science to the society. And so today I just thought the topic I can cover is microorganisms for sustainable agriculture. We know that all of us are microbiologists, so we know that it's Antony van Leeuwenhoek in 1676 discovered microorganisms, and nobody believed him, because the theory of spontaneous generation was there at that time. And later, because of the work of Louis Pasteur in France, where he showed that the conversion of grape juice to wine, a microorganism is involved. And again, the silkworms, the larvae, they die because of microorganisms. That is how the importance of microorganisms came into the picture. Now, we know that microorganisms are present everywhere. In fact, it is estimated that 80% of the biological systems, including plants, animals, and microbes, 80% of them are microbes. And earlier scientists, including microbiologists, they felt that microbes are bad creatures, causing diseases in humans, animals, spoiling food, textiles, and so on. But later, the beneficial aspects of microbes were understood. So I am going to cover the beneficial aspects of the microorganisms, especially in agriculture. And we know that at the time of independence, 1947, there was a shortage of food in our country. It was 50.8 million tons, and people had to live from ship-to-mouth existence. We were all eagerly waiting for wheat to come from US through the PL 480 scheme, so that we can make roti and then eat. So that was the situation. Thanks to Green Revolution, yielding varieties of crops were introduced. So in 1970s, the crop production doubled. From 50.8, it became 108.4 million tons. And in 1996, the increase in the food production was 200 million tons. So we had enough food to eat. So thanks to Green Revolution. But what happened for the production of high-yielding varieties? We had to use heavy doses of fertilizers and pesticides. And later, after several decades, now we are realizing that it is harming our environment, environmental pollution. How people came to know this is that nitrate in the groundwater is now much higher than the recommended level. And the pesticides have been recorded in tender coconut water. Okay? So now we are concerned about the environment and the food we are eating. And so that we take less and less of pesticides along with our food. That is why the concept of sustainable agriculture or organic farming came into the picture. So it's not only in India, but all over the world. So what exactly we do in sustainable agriculture? What is recommended? Use less chemicals and more biologicals, like compost, green manure, biofertilizers, biocontrol agents, and so on. Let us take the biofertilizers first. And we know that nitrogen, phosphorus, and potassium are the three major nutrients required by plants. Let us take each one of them. Let us take nitrogen first. We know that plenty of nitrogen is present in the atmosphere. Nearly 78% is present in the atmosphere. But plants cannot use them directly. Okay? But there are many bacteria which are present in soil which can fix this atmospheric nitrogen and make it available for plant growth. All of us, right from our school days, we are taught that legumes have root nodules and rhizobium sits inside the nodules. All of us know this. So we are very, very familiar about this rhizobium fixing atmospheric nitrogen. Now we have close related genera of Rhizobium like Sinorhizobium, Allorhizobium, Bradyrhizobium, Mesorhizobium. But there are many other organisms, bacteria, which can also form nodules in the roots of legumes and fix atmospheric nitrogen. Phylobacterium, Methylobacterium, Achromobacterium, Burkholderia, Cupravides, Devosia can also form nodules in the roots of legumes and can fix atmospheric nitrogen. How many of us know this? Because not even one of these organisms has been come out from India. All these reports are from outside countries. So there is ample scope for young microbiologists to look whether these bacteria are present in our soil and they cause nodules in our legume roots. And we know that these Rhizome can fix up to 100 kg of nitrogen per hectare per season. And now let us think of a farmer. A farmer is growing groundnut and he wants to inoculate his groundnut crop with the best Rhizobium. Then how, as scientists, as microbiologists, how we can help? So what has been done is, this is a just root nodule which shows the nodules. These nodules are pink in color because of a particular pigment called leg hemoglobin. More the leg hemoglobin, it is a better nitrogen fixer. Okay, now what we do? We want to select the best Rhizobium for inoculating groundnut. How do we go about it? We go to a groundnut field and we see here and there some plants are growing better. They look more healthy. So we presume that probably they are having better Rhizobium in the root nodules. So we bring the nodules, isolate the Rhizobium. Let us say we have isolated about 80 different Rhizobia from the groundnut field. Now we have to select the best Rhizobium. What do we do next? We have to shortlist it. So we do this plant test tube assay. So this is the plant test tube assay using large test tubes and using Ceratro as the model legume. And we do this assay and we can shortlist the plants which grow better are selected. So from 80, let us say we come down to 20. So now we have shortlisted from 80 to 20. What do we do next? We have to further shortlist it. We do this assay in the glass house called Lenore jar assembly assay. So what we do in this case is we take a Horlicks bottle. So this is a Horlicks bottle at the bottom. The top is the beer bottle. The bottom of the beer bottle is cut and inverted over the Horlicks bottle. And we take a substrate. It could be a sterilized sand or any other growth medium. And we put a cotton wick in the middle, and then it comes down to the Horlicks bottle, and we take a nutrient solution. And so the nutrients go up through the wick and give the moisture to the plant. And we sow the legume here. And so what happens? So from this assay, we looked into the growth. And then from 20, we can now shortlist to 5. Okay, the best olive. So from 80, we have shortlisted to 20. From 20, we have shortlisted to 5. Now we have to select the best Rhizobium again. We have to shortlist the best one. What do we do next? It's the microplot assay. Microplot assay is just 2 meters by 2 meters we can take and then inoculate with these 5 shortlisted Rhizobia and look for the plant growth. Again, let us say from the microplot assay, we have shortlisted to 3. What do we do now? Next, now the field experiment comes in. So the macroplot assay, and this is done all over the country. We have All India Coordinated Research Project, and they will help us to do this experiment in different agroclimatic regions and different soil types. So this is one such experiment. And so you can see the difference between uninoculated versus the inoculated. And we select the best Rhizobium, so which can be mass multiplied through fermenters and can be given to the farming community. Now we have come to that extent, a farmer can go to a shop and buy the best Rhizobium for inoculating his groundnut. So this is one of the DST project which we completed a couple of years back. This was done in a place called Palamanar in Andhra Pradesh. And you can see the difference in growth of groundnut. Okay, inoculated versus inoculated. So that is because of the lack of rhizobium in that particular soil. Now it has become a common practice. All the farmers in that region use rhizobium inoculation. And I have just included one of the results by the scientists from all over the country, where they have inoculated with rhizobium in various crop plants in chickpea, cowpea, pigeon peas, red gram. And then we have the results where up to 20%, 30% increase in yield has been reported. And the other thing which I want to introduce here is another type of nitrogen fixing organism. We always think the rhizobium fixers are form nodules only in the root, but some of them form nodules even in the stem. They are called as stem nodule bacteria. So the organism involved is the Azorhizobium colinodens, and the plant in which it forms the nodule is Suspenia rostrata. It is a green fruit plant. So the paddy growers grow this particular green manure plant and then they puddle it into the soil, then transplant rice. So this is a very common practice with paddy growers. Okay, now see, this is the plant, Suspenia rostrata, and you can see the stem nodules. So we have the advantage, it forms the nodules both in the stem as well as in the root.
अरे छोड़ दो यार, आगे जल्दी छोड़ दो नौटंकी रहने दो यार, हम ही परचेज कर लेंगे, रहने दो मत करो। अब उन्हें बोलो आ जाओ भाई इधर आके।
Please mute yourself.
हर बार यही होना
disturbance, please.
Hello, Dr. Patar. Please unmute your
100-150 रुपए का
Dr. Patar, please unmute your mic. Close your mic.
Okay. Now, other thing, another plant which forms the nodules in the root is not by rhizobium, but it is by Actinobacteria, actinomycete, as it was called earlier. The name of the organism is Frankia. It forms nodules in Casuarina. We see Casuarina growing in sandy environment very luxuriantly. That is because it forms such big nodules. The nodules can be as big as a table tennis ball, so big as that, and fixing atmospheric nitrogen. And this is one of the experiments done by us. We can see the difference in plant growth because of inoculation with Frankia. And that is the symbiotic nitrogen fixing bacteria, which forms the nodules. And the second category of nitrogen fixation is called associative symbiotic nitrogen fixation. Here, the bacteria will not form any nodules, but it remains in soil and it penetrates the epidermis of the plant or the outer cortical cells, and they fix atmospheric nitrogen. So the classical example is Azospirillum. And another organism, again, a symbiotic nitrogen fixer, is Gluconoacetobacter diazotrophicus. It's a sugar-loving bacterium, a saccharophilic bacterium. So you can imagine which type of plants they are present. They are associated with plants like sugarcane, sweet potato, sweet sorghum, and so on. And again, a lot of work has been done on Azospirillum in our country, and you can see the percentage increase has been reported by different workers. And the third category of nitrogen fixing organisms are the free living nitrogen fixing organisms. They live in soil, colonize the rhizosphere, and they do not enter the plant. So they fix the nitrogen which is taken by the plant. The classical example is Azotobacter, and which is very, very common in most of the soils. And people from different parts of the country have reported the increase in yield because of inoculation in various crops. And another free-living nitrogen-fixing organism are the cyanobacteria, earlier called as blue-green algae, like Nasta, Cannabina, Tolipothrix, and so on. And they are all photoautotrophic organisms, which is utilizing sunlight as the energy. And heterocyst in their filament is the site of nitrogen fixation. I have just taken one example here, where in Madhya Pradesh, 17% increase in yield in rice has been reported because of inoculation with cyanobacteria. And this shows the heterocysts which are present. And this is a slide I could make in Tamil Nadu, where the farmer is multiplying cyanobacteria in his own field. So what he does, he makes a trench, puts a polythene sheet, and then adds some water and inoculate with the starter culture. And in about 3 weeks' time, he has plenty of inoculum of cyanobacteria, which he introduces in the paddy field at the time of transplanting rice. So this is a very South India. And another organism is Azola. Azola is a water fern harboring the bacterium Anavina azole in leaf. So this water fern also can be multiplied by the farmer the same way as I told you for blue-green algae. And it can be added on to the rice field at the time of transplanting rice. It's again a very, very common practice by farmers. And so this is how the water fern Azola looks like, a single organism, a single plant and a colony of it. And here again, in Kanyakumari district of Tamil Nadu, when I had a visit, I could see that the farmers have raised the Azola inoculum, and I was surprised to see that they use it for feeding chicks, for feeding sheep and cat, and also cows. And I asked them what the benefit they are getting. What they said is they increase growth of the chicks and as well as the sheep, and they get better milk yield. So this is slowly getting into the other parts of the country. So which is started right in the right south of India in Kanyakumari, it is coming up. Many people have started using this. Coming to the next most important plant nutrient, phosphorus. You know, most of our soils are tropical soils, and most of the tropical soils are deficient in phosphorus. And when a farmer adds phosphatic fertilizer to the soil, what happens? Nearly 75 to 80% of them are converted to an unavailable form, which the plants cannot use. In acidic soils, they are converted to iron and aluminum phosphate. In slightly alkaline, saline soils, they are converted to calcium phosphate, which is not available for plant growth. But in the soil, we have many phosphate solubilizing microorganisms, both bacteria as well as fungi, like Bacillus polymixa, Pseudomonas striata, Aspergillus avamore, and so on. So they produce organic acids and convert the unavailable form of P to an available form. So plants take phosphorus in the form of H2PO4 ion. So they convert and release this H2PO4 ion so that the plants can take. And again, in our country, there is plenty of cheaper source of phosphatic fertilizer, that is rock phosphate. And many experiments have been done using rock phosphate and then treating them with a phosphate solubilizing organism and then looking into the growth. I've just included one slide, how we just select the best phosphate solubilizing bacterium in the laboratory, looking into the zone of solubilization in Petri dish. And we have just one best fungus and one bacterium, which are phosphate solubilizers. And this is the result of the experiments done by various scientists in our country using phosphate solubilizing organisms, and the percent increase up to 56% in cabbage has been reported. So this is very, very interesting. Coming to another group of organisms, the arbuscular mycorrhizal fungi. And so these fungi also help in the uptake of phosphorus for plants. And a lot of studies have been done And here you can see the difference in growth because of uninoculated versus inoculated. The left one is uninoculated, the right one is inoculated. So lots of reports are throughout the world, including India. And how do they help plant growth? If we just take one example, let us take cotton. You have uninoculated versus inoculated. You can see the difference in the dry weight, significant difference in dry weight, and see the difference in the uptake of nutrients like phosphorus, zinc, manganese, and so on. So they help in the uptake of nutrients, which do not move freely in soil solution. We call them as immobile nutrients. So they help in the uptake of immobile nutrients. The next slide will explain you better. So this is the plant root, and we all of us know that plants take the nutrients through the root hairs. So the root hairs are not very long. So as soon as the phosphorus is taken from the root hairs, there is no more phosphorus left in the root hair region. So we call it a phosphorus depletion zone. And what the phosphorus the root hairs have taken, there is no more phosphorus left. But here the mycorrhizal fungi play the more important role. They travel much longer distance compared to the root hairs, scavenge a larger volume of soil, take the phosphorus and drop it into the plant, thereby short circuiting the nutrient. So this is one of the major methods by which it helps plant growth and uptake of nutrients. They also have a synergistic interaction with the nitrogen fixing organism. What do I mean by this? That means when a plant is inoculated with Rhizobium as well as the mycorrhizal fungus increases nodulation and nitrogen fixation by Rhizobium. And Rhizobium in turn increases colonization and uptake of phosphorus by the mycorrhizal fungi. That's why there is a synergistic interaction. Again, these mycorrhizal fungi help in the biocontrol of plant pathogens. So the second plant is a groundnut plant infected with a fungus, Sclerotium ralsei. The third plant is infected with the fungus pathogen and also inoculated with the mycorrhizal fungus. You can see the difference in growth, which very, very clearly shows that it helps in alleviating the plant pathogenic fungus or the severity caused by the plant pathogen. The mechanism has been worked out. We know that it is because of the higher concentration of orthodihydroxyphenol in the mycorrhizal roots, which makes the plants, protects the plants against the pathogen. This is another ICR project which we completed very recently, and we can see it helps in the drought conditions. You can see this is a soybean plant under water stressed conditions, and here you can see the plants under water stressed conditions but inoculated with the mycorrhizal fungi. And just summing up the mechanism of improved plant growth due to mycorrhizal, and it is because of increased nutrient uptake of less mobile nutrients, synergistic interaction with other soil organisms, protecting plants against pathogens and protecting plants against drought. And they also produce growth promoting substances like indolactic acid, cytokinin, gibberellin, and it's a cumulative effect that makes the plants grow. And they also help in reducing the amount of fertilizer we add. So here I just included one example of chili, where the recommended level of phosphatic fertilizer is 75 kg per hectare. And here we have reduced it by half, inoculated with various mycorrhizal fungi. And this is a field experiment where you can see the yield per plot. You can see with the recommended dose of fertilizer, it is 1.81 kgs. And here it is with this mycorrhizal fungus, with 50% reduction in phosphatic, it is 2.14. So this we have done in several crop plants, and now everyone knows that it helps in phosphorus nutrition and reducing the amount of phosphatic fertilizer. Another type of work, what we did is developing a microbial consortium. And this is uninoculated Acacia auricularis. The second one, inoculated only with the mycorrhizal fungus. Now we have a microbial consortium, mycorrhizal fungus plus some plant growth promoting microorganisms. So you can see the difference, which very clearly shows a microbial consortium works better than inoculation with a single organism. So this is a DBT project again, which we completed a few years back. And where in and around Bangalore, we have a severe problem in tomato. That is a wilt disease complex, which is caused by a fungal pathogen, bacterial pathogen, and a nematode pathogen. So that is why it is called as wilt disease complex. So in this project, we have developed a microbial consortia consisting of a mycorrhizal fungus, Pseudomonas fluorescens, and Poconia license, and which takes care of all the three pathogens. You can see the difference of the seedlings raised in fruit trees. We went up to the planting them in wilt-sick soil. Again, it protected them against, and we would get better yield in the microbial consortium inoculated plants. And here's another experiment which we could do, and with chili as the crop. And here is the 100% nitrogen, phosphorus, and potassium (NPK) fertilizer. You can see the yield. It is 1,451, and with 50% of the reduction in all the three fertilizers, NPK with the microbial consortium, you can say significantly similar yield, whereby the farmer can reduce the application of fertilizer by 50%. And this was a field experiment which was done by one of my PhD students. And so you can see this field experiment. And of course, this is an Indo-Swiss project where we have right now, where we are trying to see whether the mycorrhizal fungus, when intercropping with a deep-rooted plant like red gram with ragi or finger millet, whether under drought situations, whether the mycorrhizal hyphae can take the water from the deep-rooted plant and then irrigate the shallow-rooted plant, which is called as bioirrigation. So we are just having this project, which is going on at present. And coming to the third major nutrient, that is potassium. The first report of the potassium solubilizing bacterium came from Orissa, and the bacterium was identified as Pretoria aurantia, isolated from banana rhizosphere. Of course, now we have various other organisms which can also mobilize potassium for plant growth. What has been studied so far shows that a farmer can reduce the application of potassium fertilizer by 50% through inoculation with this bacterium. Another category of organism is the plant growth promoting rhizome microorganisms. And when we talk of the literature and go through the literature and see what has been done globally, two organisms come into our mind. One is Pseudomonas fluorescens, where extensive studies have been made throughout the world, including India, on Pseudomonas fluorescens as a plant growth promoting organism, PGPR. The next organism which has received some attention is Burkholderia cepaceae. But look at the international literature. Methylobacterium, Azoarchus, Pantia, Penibacillus have been listed as the best PGPR. But we have very little work in our country on these PGPR. Of course, Methylobacterium species, some work is going on in Tamil Nadu Agricultural University, especially the Madurai campus, where they have shown extensively that it helps in plant growth. Whereas the last three, very, very little work, ample scope for young microbiologists to work on these organisms. So here, again, I would like to say that the PGPR work, we had a scientist from Pantnagar. She got a DST Women Scientist scholarship. And so through this project, she came and worked in my lab. And the crop she worked was French bean. And you can see that of the various PGPR she studied, and Pantia agglomerans proved to be the best PGPR for inoculating French bean. And she did the lab work, then took it to the glass house, then took it to the field, finally ended up with the farmer's field. So now again, this particular technology is followed by the farmers growing French bean in and around Bangalore. And another student who came from Tamil Nadu, and for his PhD, he worked on chili and selected the best PGPR, and this is Bacillus licheniformis. Again, all the field experiments, including the farmer's field, And now this has become a common technology by chili growers. And coming to a different aspect, the Karnataka Sugar and Distilleries Association had a problem, and they came to us and said for decomposition, they wanted us to help them. What happens in sugar industries and distilleries, after crushing the sugar cane and whatever the bagasse they get, it takes a long time to decompose. They came and said whether you can help us using some lignocellulosic organism where it can decompose much faster. We selected 4 different fungi: Phenorrheic Chrysosporium, Trichuris spiralis, Trichoderma viridae, and Pleurotus kaju, which is a mushroom organism, but very good decomposer also. So a combination of this was used for this. And this of the windrows, how the sugar industries or distillery industries make these windrows for decomposition. And you can see the aerotiller, which we suggested them, where we can have a mixture of these 4 fungi and then introduce it into the windrows and then mix it so that the fungi get into the windrows and then help in decomposition. Now, this is a close-up, how it is introduced. And now you know that 27 distilleries and sugarcane industries in Karnataka are now following this. And now the decomposition takes place in 2.5 to 3 months. So it's a big contribution, and which everybody likes, and it is spreading to the other states also. And coming to the biocontrol, and we know that insect pests and diseases are the major problem. So whether microbes can help as biocontrol agents to protect the plants against this. So I'll take one example, pigeon pea, which is red gram. And the serious problem in pigeon pea is the wilt disease caused by Fusarium woodum. And after screening several biocontrol agents, what has been found is the Trichoderma viridae is the best biocontrol agent for controlling the wilt disease of pigeon pea. So here we have various crop plants, chickpeas, soybean, brinjal, cabbage, and the major disease of this. And you can see the list best biocontrol. If looking into the third column, you can see Trichoderma dominates. So Trichoderma is a very good biocontrol agent. And again, it is available in the market. A farmer can go and buy Trichoderma and use it in his field. And just to how we test the antagonism in the laboratory. And again, another problem, in and around Bangalore, the solanaceous crops are attacked by the root knot nematode, which is a very serious problem, not only in vegetables, variety of croplands. So again, how we can help in control of these root knot nematodes. And we have 2 fungi, Bacillomyces lelacinus and Pucania chlamydosporia, are very effective in controlling root knot nematodes. And coming to the biocontrol of insect pests, taking cotton as the example, and we know bollworm is a major problem. The best biocontrol agent is the Bacillus thuringiensis, and from which we know that we has been. And how this insect is what are the major biocontrol agents. So this is, as I told you, is one by Bacillus thuringiensis. I just made it at 130 dish having Bacillus thuringiensis.
1 minute, please.
It is sprayed.
Please mute your mics.
Killed, and this is
Please
how you see
mute
the
your
dead
mics.
larva in the field. And other fungi which are very effective biocontrol agents are Beauveria bassiana, which is called as the white muscardine fungus. When you see this beetle has been infested with the fungus, the whole insect turns white. So that is why it's called as white muscardine fungus, and it dies. And another fungus, which is a very effective biocontrol agent, is the green muscardine fungus, Materiasium anasophiliae. And you can name why it is called green muscardine. The caterpillar, the grasshopper or any other insect attacked by this turns green, and that is why it dies. And that is that's why it's called as green muscardine fungus. And coming to the viral pesticides, and we know that the major problem, insect problem in our country are the 2 insects. Helicoverpa varmigera and Spodoptera litura, which attack a variety of crop plants. So the best method to control these two insect pests is the nuclear polyhedrosis virus, which works extremely well in controlling these insects. And here I've just included one slide showing a larva killed by the nuclear polyhedrosis virus. It is very, very interesting to see how the larva, when it is infected, it just goes to the top of the plant. Even a tree, it goes to the top of the tree and then hangs upside down, the head down, tail up, and dies. So just looking at the way in which the larva has died, you can guess that it has been attacked by the nuclear polyhedrosis virus. Okay, coming to the biocontrol of weeds, lot of projects in our country has been financed, but unfortunately, till today, we do not have one biocontrol agent effectively used for biocontrol of weeds. And one which showed some promise was the Puccinia spizanginae, which is a rust fungus against Mecania weed. And Mecania micrantha is a scientific name, which is a problem in plantation crops and forests. So this Cabby did the initial experiment, then it transferred it to NPGR and NPPGR, and then they released it in 2 states. They released it in Assam and Kerala in the coffee plantations and tea plantations. And Kerala people said, first year there was little promise, the second year it did not work. Assam people said, even in the first year it did not work. So that is why till today, we do not have a one microorganism which can effectively use for controlling weeds. And this is Mecania micrantha weed, and this is the fungus which was used. And we do the training sometimes for people who ask for, sometimes farmers, sometimes the scientists. And let us see how biotechnology has helped microbiology. I'll just take a couple of examples. And you know that herbicides or weedicides are used by the farmers to control or kill the weeds in the farm, in the cropping area. And glyphosate is a very common herbicide that is used by the farmers, but some crops are sensitive to this. For example, tobacco. Tobacco is sensitive to this particular herbicide because that also will be killed. So there are bacteria which are resistant to glyphosate. So the genes from this resistant bacteria have been transferred through protoplast fusion and to tobacco. Now we have glyphosate resistant tobacco. Now the farmer can raise glyphosate resistant tobacco and use glyphosate for controlling weeds in the tobacco field. And another example I would like to give is, we know that Bacillus thuringiensis produces toxins against Lepidopterous insects. And this gene has been transferred to the common rhizosphere bacterium, Pseudomonas fluorescens. And the farmer can treat the seeds with Pseudomonas fluorescens before he can sow. And now what happens? It colonizes the rhizosphere. And the black cutworms, they try to feed on the roots. So they nibble the roots. When they nibble the roots, they also consume Pseudomonas fluorescens containing the gene from Bacillus thuringiensis. And they consume and they die. So these are two classical examples where biotechnology has helped the microbiologists. And nowadays, a lot of importance is given to nanotechnology. Earlier, people were using plants for producing nanoparticles. Now we know it's much more economical to produce nanoparticles using microbes. And they have a lot of significance in the rapid diagnosis of diseases, development of vaccines, control of environmental pollution, preservation of food. They act as antimicrobial agents and so on. And coming finally to biodiversity, where a lot of importance is given at present globally. And we talked so much about microbes, how they are useful in agriculture. Now, how much of the microbes existing in nature do we know? It is so sad. We know only 3% of the microbes existing in nature. 97 of them we do not know. Okay. Now, the Global Committee on Biodiversity, they they had a discussion on this, and they questioned, where are these 97% microbes hiding? Okay, and sorry, and they came up with some places, environment where they may be present. They have just expressed this, not definitely they are present there, probably they are present. Okay, so they are hypersaline lakes and soils, thermal environments including hot deserts, mangroves, swamps, brackish waters, salt marshes, beaches, estuaries, surface intestines, fecal matter and nests of insects and animals, surface of plants growing under extreme environments, ocean floors, and environments saturated with sewage effluents and pollutants. So these are the areas where the 97% of our microbes may be present. So it is a chance. So there is ample scope for young microbiologists here to try to isolate newer bacteria, new fungi from this area, and then see how they are impaired. Bioprospect them, maybe a better nitrogen fixer, better enzyme producer, okay, better antibiotic producer. So there is ample scope for our young people who are here. And in this connection, I would like to give an example. We know that through nitrogen fixation, let us say rhizobium, nitrogen fixation, the atmospheric nitrogen is converted to ammonical form of nitrogen. And if you see all our textbooks till recently, what do they say? But plants do not prefer to take ammonical form of nitrogen. They prefer nitrate form of nitrogen. Then how this ammonical form of nitrogen is converted to nitrate? It's a two-step process. Ammonical form of nitrogen is converted to nitrite by nitrosomonas, and nitrite is converted to nitrate by nitrobacter. So it is a two-step, nitrification is a two-step process done by two different and now recently from the University of Maryland, one scientist by name Santaro has discovered a bacterium and he has named it as Nitrospira. And he found that it can convert ammonical form of nitrogen directly to nitrate. That is single step process, which brought him a lot of credit. So I am sure that the young people here can isolate such new organisms, discover them, bioprospect. There is ample future. And I wish the young people here grand success and a bright future. Thank you very much.
It was an excellent lecture by Dr. Joseph. I really enjoyed the lecture very much.
Thank you.
It had very good PPTs and very colorful green slides, which I really appreciate. Now it is question time. So you can ask questions to Dr. Joseph.
Yeah, or you can put in the chat box also. That may be.
Yes. Yes. Please ask question to Dr. Joseph Bhagyaraj.
Yeah.
Good afternoon, sir. So please, may I ask the question?
Yeah, please.
Generally, we source the potential biofertilizers from soil. But there is a school of thought which says that we should source it from the plant microbiome, the seed microbiota, or the phyllosphere. So what is your take on this? Thank you, sir, and a very nice lecture.
Okay, just I could hear you properly. That's why I'm asking the young person by this side. Yeah, sure. You can get it from plant also, not necessarily from soil. It can come from plant. Yeah, sure. Why not? From phyllosphere, okay, phylloplane, everywhere. Yeah.
Dr. Joseph. Sir, may I ask some questions? One question.
Yeah, sure. If you can put it in
chat
box, it may be better so that I can see and then answer.
I am from Bundelkhand University, Jhansi. That one is part of Uttar Pradesh. I'm working on I'm working with some students on mycorrhiza, particularly on pulse crops. That one is basically isolated from the rhizosphere of chickpea and lentil. We are facing some problem in identification. So we are not able to recognize. We are having a scarcity of manuals even. Can you suggest me how we can go for identification, or how we can from where we can take help for that?
The identification of fungi, I think it takes quite a bit of time. And I was doing it earlier. Now I stopped doing it because of too many commitments and all that. But I can suggest somebody who can help you out. There is one Dr. Lakshmipati. I will send, if you can send me a mail or a message, I will give you the address and contact of Dr. Lakshmipati, who was my student, who is now in Andhra Pradesh Agricultural University. He may be of help to you.
Dr. Anil Kumar Garg sir was in Kafri, Central Agroforestry Research Institute in Jhansi, but he got retired. He shifted in Haryana. Earlier he was helping me, but now I don't have any kind of assistance. I humbly request you, can you share me email on chat box, sir? Then I can send the mail and pictures even to get your assistance for identification.
Okay, fine.
Dr. Manjeet Singh.
Dr. Yusuf, good morning. So it was an excellent talk because I'm primarily agriculturist and from a totally different discipline. By degree, I am a geneticist who worked on mushroom. So my questions can be slightly odd. One thing that, yes, there was the excellent work by many workers where we reported up to 30, 40, 50% increase in the yield. But repeatability, I think, becomes a big issue because we are not able to capitalize all this good work for the development of agriculture. Because I am not aware of many technologies. Because we have been hearing about rhizobia from times immemorial. Many universities tried to prepare the consortia. But still, we need to take the technology to the second level so that we fine-tune it to the level that everywhere. I think one reason is that the soil type, soil temperature, soil moisture, nutrient status of the soils, and most importantly, which we don't talk much, is the microbial ecology of the soil. Differs from place to place, and I think that is a reason that maybe we are not able to repeat it. And that's, I think, the beginning that instead of focusing the research only at one location, we have to go multi-locational with our testing. Secondly, I think the second comment was regarding your PSM. I think that is a really good work. And I know my colleague, Dr. Tarafdar at Kazri, Jodhpur. He did a lot of work on this phosphate solubilization. Then he shifted to nanotechnology. I wish that it is such a beautiful discipline, microbiology, and we have got such good cultures. The time has come because the country is now gearing for chemical-free agriculture. Even our PM is trying to promote chemical-free agriculture. So the stage is ripe that now instead of lab, we should shift more to the field and commercial technologies. I think these were a few comments and not exactly the questions. And thank you for the excellent talk.
Okay, thank you. Yeah.
I have one small query. Can I talk to you?
Yeah, please.
A new AM fungus has been named after your name. This is a great honor to you. Please tell us more about this. The AM fungus.
Okay. I think it was made, it is from somewhere in Allahabad. This fungus was from Allahabad soil is what isolated. And one Dr. Vinay Mehrotra, who I happen to be his PhD thesis examiner. So he liked me, perhaps he likes me very much. So it has been named after me.
Very good. It's a very nice thing. I noted that. It is after your name. So any more question? Any more question? Okay, last question I'll ask you, Dr. Joseph.
Yeah, please.
How did you start working on mycorrhizal fungi?
Mycorrhizal fungi?
Yes.
Okay, it's a very interesting question.
Yes.
I had a Fulbright fellowship in the year 1976 or 1977, I think. So it was in the University of California. And I was to work with the, suddenly I'm not getting, the person, the AM, the annual review of microbiology, chief editor, Dr. Mortimer Starr. Okay, so I was to go and work in his lab. And the thesis on which I was supposed to work, my postdoctoral work or a Fulbright fellowship work was transfer the nitrogen fixing gene from Rhizobium to a saprophytic bacterium, Erwinia herbicola. So that was the problem in which I am supposed to work. So I landed in University of California, and Dr. Starr introduced me to the staff, and I started working. The next lab was working on mycorrhizal fungi. So I used to peep into the next lab. Somehow I felt that what the people were working on mycorrhiza is more interesting than what I is supposed to do. Okay. And so I came back and told Dr. Mortimer Starr, is it possible that I can shift to the next lab and work on mycorrhizal fungi? Because I feel that when I go back to India, that will be more useful to the farming community. Then he said, you come on a Fulbright fellowship and the head office is in Washington, D.C. You call the Washington, D.C. people, and if they don't have any objection, in the other mycorrhiza professor has no objection, I will not have any objection. So I called the Fulbright office people, and that lady took the phone and said, if both the professors have no objection, we have no objection. So that is how I shifted to the next lab. And when I was to complete the 1 year's Fulbright fellowship on December 31st of that year, and sometime in October, the professor told me, if you want to stay back in US, we can take you on a contract basis for 5 years, and another project on mycorrhiza. I was perplexed at what I'm supposed to do. I was married at the time, so I called my wife as well as my parents and asked what I am supposed to do. And they said, we leave it to you, do what you feel. Then I talked to my Indian friends who were in the campus, in the University of California campus. And they strongly advised I should return. The reason they said was, number one, we are sticking here because we couldn't get a good job in India. And we came for a postdoctoral fellowship and we are continuing here. But you are already an associate professor in the University of Agricultural Sciences, so you should go back. I felt what they said was right. So that is how many people said I am a fool, leaving the offer and then coming. So anyway, when I came back, going through the literature, I could see that practically nothing has been done on mycorrhizal fungi, except a very few work on ectomycorrhizal fungi on forest trees from Dehradun, Forest Research Institute, Dehradun. So in crop plants, practically nothing has been done. So we have to start from a scratch. So everything was something very new. And we did not know that even these fungi exist in our soils. Okay, so that from that extent, we have to start. And finally, what happened was, when the work was started, I had wonderful students working with me, and we have to contradict many of the findings which came from the Western world. The Western world said mycorrhizal fungi never colonize aquatic plants. We reported the world first report that it colonizes aquatic plants. And other thing is, the Western world said that the mycorrhizal fungi sporulate in summer, and we have to say that it sporulates in winter. All these things, which was something different from what has been reported, that brought. Another thing is mycorrhizal fungi are not host specific. So we cannot select a particular fungus for inoculating a crop plant. So later we said that it is, though it is not host specific, they have host preference. So this, nobody, and I agreed, neither the US scientists nor the European scientists. So a big project came from United States Department of Agriculture to me just to prove this hypothesis. So many students were benefited. Now we know that everybody talks that we can select the best mycorrhizal fungus for inoculating a first crop plant. So I think probably all these controversies brought name and fame to our institution.
Very nice. Very nice.
Final question, if I'm allowed.
Okay, one question before we conclude.
Yes, sir.
You talked about that sugarcane bagasse, sugarcane residues.
Yeah.
In mushroom, we are using a lot of sugarcane bagasse as a substrate for growing button mushroom.
Okay.
And we allow the natural microflora to take that, mostly thermophilic microbes, which help to degrade. And no issues at that stage, but our problem is that once we grow the mushroom, what is left, we use the word SMS, spent mushroom substrate. There have been few reports here and there about microbial mixtures which can help us to degrade that spent mushroom substrate and convert it into a good fertilizer, good manure, very easily. But I'm not really sure whether these things work or not, because it is already fully colonized by the fungi,
mushroom fungus,
by the mushroom fungus. And whether those microbes will be able to compete, or are you aware of some microbial mixtures which work on this spent mushroom substrate, the term which we use in our literature?
No, sir, I do not know about it, but somebody from Tamil Nadu, I think somewhere near Chennai, they approached me just a few days back and said whether the same question what you asked, they asked me. And I said, I really do not know. Probably it may be better to try these consortia, what we developed earlier for decomposition. And this is agaricus, no? So in that case, we have to try. So it may work also.
I have done no work on this, but there are so many claims by the private sector people. Where sometimes I feel that they are trying to befool the people, because even naturally, if you just make the heap and turn it regularly and go for what we call aerobic fermentation, then you succeed in getting a good compost within a month or so. At times, I think the natural microflora is the best microflora, because the nature establishes its own balance. But we impose on the nature, I'm really not sure whether it will work in the long run or not. And that's where I think mentioned the word of microbial biodiversity. We have got a biodiversity at the top level, but what happens inside the soil, that biodiversity is equally important. And any work done in isolation without giving proper consideration to that microbial biodiversity will only be half of the story. Thank you.
Right. So it will be very interesting to try this, actually, whether the mushroom waste that's coming out can be decomposed.
We already used one of the mushroom. Pleurotus sajarkaju is basically a mushroom.
Pleurotus is very effective, very good also.
Lignocellulose degradation, for that it is highly effective. Thank you.
Thank you very much. Now I call upon Dr. Avneet of Medical College, Chandigarh, to give us vote of thanks.
Thank you so much, sir, for inviting me, and thank you everyone for your kind questions. And I'm sure we all can agree that knowledge is which one can experiences and wonderful insights have shown us all a new and different perspective of things. We're extremely grateful and honored for giving us your time, sir. I would also especially like to thank the President and the team of Chandigarh MBSI for putting hard work and effort. Thank you so much for joining us today.
Okay. Thank you. Thank
Thank
you
you very much.
Thank you, everyone.
Thank you.
Thank you, sir.
Okay.
Thank you so much, Dr. Avneet.
My pleasure, sir. Thank you.
Thank you.
Wonderful lecture, sir. Very nice lectures.
Okay. Thank you very much, Dr. Swaranjit Singh and Dr. Deshmukh. And very, I
Thank you
really
so much.
see
We
you.
really enjoyed your lecture.
Thank you.
Bye then.
Bye.
Bye.